Hello and welcome back again to another video tutorial of Roy's Academy. In this video tutorial I am going to discuss about the base pairing model of the DNA structure. Okay, how the base pairing happens and what are the necessary formulations that were proposed by the scientists, which were brought forward in the limelight in order to understand the sole structure of the base pairing, that how the nitrogenous bases are pairing with each other and how they are present in the nucleotide as per a particular formulation basis. So those formulations were being proposed by the scientists in order to understand the DNA structure in a more simplified manner. Okay, because there are innumerable number of bases. Obviously, if we go to count the bases in manual mode, it is not possible, and obviously in the gene editing technology or in laboratory based methods also, it is in the wet lab method, it is also not possible quite often. But if we have certain formulations in order to understand that how many we can predict, actually, we can understand or also assume that how many total number of bases might be present there. Okay, through some computational basis or dry laboratory basis. If we from the wet lab, if we bring the sample or subject it to dry laboratory basis, there are certain other techniques, bioinformatical techniques, computational techniques and all, detection methods and all. We can understand that the DNA is how much is the weight of the DNA and how many base pairs might be present there. And from those formulations we can calculate that what would be the percentages of each of the nucleotides present within that DNA. Okay, so let's see that how we can understand the base pairing model of the DNA structure. Okay. This is what the topic of discussion is. Now, before going into the detailed explanation that how the base pairing is happening, let me draw the diagram of the nucleoside as well as the nucleotide firstly. Okay, so we have the nucleoside. First, let me draw the ribose sugar. I am drawing the structure of DNA, that's why it is deoxyribose. The second carbon will not have the hydroxyl radical, whereas the third carbon will have, okay, H and CH2OH. Now, let me draw the phosphate group present here. OH, the phosphoric acid, double bond O, OH. Again, along with this OH, there would be this OH bond present, which will be linked to the carbon of the preceding ribose sugar. Okay, so this is what one strand of the DNA molecule is. Now, along with this strand, if I draw the structure of the nucleotide, this heterocyclic nitrogenous base is linked with this first carbon, right? In case of purine, always remember, in case of purine base, that is adenine and guanine, they are heterocyclic rings. The ninth, in the ring configuration, the nitrogen which is present at the ninth position of the ring forms the bond which is called N-glycosidic bond with the first carbon of the ribose sugar in case of the purine base, the ninth nitrogen. And in case of pyrimidine, the number of nitrogen is, the position of the nitrogen is the first one. So let me draw a purine base. Let it be the adenine. Okay. Let this be the adenine. I'm drawing the adenine base here. NH. Okay, sorry. This will go off because H2O, the molecule of water will be warded off. C double bond N. C So this is what roughly an adenine base looks like. Okay. This is what roughly an adenine base looks like. Now in this case, let me shorten it out. Then it would be easier to draw the base, the purine base, pyrimidine base along with it here. I think it's quite shortened only. It would be enough to draw. Yeah, it would be enough. So this one is the adenine base pair. Adenine heterocyclic nitrogenous base. Now let me number the positions of the ring. Okay. Always remember, in case of the purine, the numbering starts from this larger ring. Okay. The numbering starts from the lone nitrogen, which is not having any other electron pair or electron sharing with any other radical or atom. So this is the lone nitrogen which is having double bond with this carbon, and there is a partial double bond characteristics present with this carbon atom. Okay, as per the amino acid rule. So this is the first nitrogen 1, let it be 1, and this one is second position 2, this is 3, this is 4, this is 5, and this is 6, and this one is 7, this position is 8, and this one is 9. Now in the original ring, without this bond formation, in the original ring there was an hydrogen present with the nitrogen atom here, and here the carbon was having OH. What happens is this hydrogen and this OH is being dropped out or removed, which helps in removal of the water and formation of this carbon nitrogen N-glycosidic bond. So that's why this is how the bond formation is happening, the N-glycosidic bond formation. This is how it happens. Okay. And now in this case also, and in this case also in the phosphate, here also if you consider this hydroxyl radical, this hydrogen and this OH would be removed. Okay, this will be minused or removed. So the bond will form in this way. This oxygen will form bond with the carbon present here, and this OH will be totally removed. OH will be totally removed. This is how the bond formation is happening. And here also this oxygen will not form this hydroxyl radical along with this hydrogen present with this hydroxyl radical of the phosphate will be removed. So here it will form the bond. Oxygen will share two of its electron, one with the carbon and another with the phosphate. And here also when the hydrogen or the proton is being generated, it becomes minus, and here already 2 electrons are paired with the phosphate atom, the phosphorus atom, so there is no unpaired electron, so only this oxygen would be having the extra negative charge. Okay, and this is the first carbon, this one is the second carbon, this one is the third carbon, this one is the fourth carbon, and this one is the fifth carbon, which is having the phosphate. Here also this one is the first carbon, this one second, third, fourth, and this one is the fifth carbon. So if I erase I remove the water from here also, it will have the same configuration. Okay, it will have the same configuration, and here it would be different. If I consider this as the ultimate end, so here oxygen would have an extra electron, here also oxygen would have an extra electron, because there is no other atom which is sharing this unshared or unpaired electron, unbonded electron of the oxygen. Now this is all about the adenine base pair which I have drawn here. Now let us consider the guanine. Adenine and guanine are the two base pairs of the purine bases, as I have mentioned. In case of the guanine, the things are quite different. In case of guanine, if I consider, then in the same way it will be like this. Nitrogen carbon is having the N bond along with the CH would be here, along with the CH bond will be present here along with nitrogen, and here again it would be the carbon, here again it would be the carbon atom present, here again it would be the carbon atom present. Okay, and along with this there will be a carbon atom which will be having a double bond O, okay, and unlike this there is a certain difference here. Here it would be the nitrogen atom present, and here again it would be a carbon which would be having a NH2, and here there would be another NH molecule which is linked with this carbon. Okay, so this is what the structure of the guanine would be. Okay, the guanine heterocyclic rings looks like this, and in this case let me name this ring also. Okay, this is the first nitrogen, this is the first position, second, third, fourth, fifth, sixth, like this, 1, 2, 3, 4, 5, 6, and here it is 7, 8, 9. Okay, this is what the representation of the ring is. Now this is about one strand. Consider this as the 5 prime. This one is the 3 prime end, because this third carbon is having the lone OH which is called the tail, and the fifth carbon is having the lone phosphate which is called the head. So this is the 5 prime head. Now let me consider the anti-parallel strand. Okay, in case of the anti-parallel strand, if I consider, again let me draw the anti-parallel strand in this fashion. Okay, now let the antiparallel strand be present here. C, double bond C, C single bond C, C, O, again here it would be C and O. Now here the things would be same like it is only. It was there, present there. Here it would be this is the first carbon, this is the last one. So here it would be H. Here it would be OH, first, second. Here it would be H, here it would be H, in this position here it would be H, and here it would be H below, and along with this carbon there would be the phosphate linkage P, double bond O, single bond O minus, again here it would be O. This would lead to the this would lead to another carbon. Let me draw it in a proper manner so that it would be easy to understand. Okay, so in this anti-parallel strand, this one is the fifth carbon, fifth position. In this case, oh sorry, I forgot to write the CH2 molecule here. Here it would be a CH2 molecule. That CH2 will join with O, and that will join with P. Here also there is a CH2 present, CH2, that is joining with O. Now this one, let me name this one also, this ring also, 1, 2, 3, 4, 5. 1, 2, 3, 4, 5. Okay. Now this is the fifth 5' end, and this one is the 3' end. This is anti-parallel. Okay. Now let me write the hydrogen bonds also here. H, H, H. Okay, this is what all about. Now, in this anti-parallel strand, we also have the pyrimidine bases. Okay, in this anti-parallel strand, we have the pyrimidine bases. Now, if I draw the pyrimidine base here, it would somewhat be like this. This carbon is having the bond with nitrogen here. The nitrogen will have the nitrogen here will have the bond with the carbon again. Carbon present here, carbon, which is having a double bond O, and that carbon will have again a bond with the nitrogen here, and that nitrogen here will again form bond with the nitrogen that is present here, and that nitrogen will form a bond with CH molecule present here. That CH will form a double bond with another CH, which is again forming a bond with this nitrogen. And now this nitrogen present over here is having this nitrogen present over here is having two hydrogens. Okay, two hydrogens. This is what the bond of cytosine is. This is what the bond of cytosine is, and the naming in case of cytosine starts in this way. This is the first carbon, first position. This is the first one, because in case of pyrimidine base, the first carbon is forming the bond with the first nitrogen. This is the second one position, third position, fourth, fifth, and sixth position. Okay. Now the bond formation that is happening somewhat like this. This oxygen is forming bond with this hydrogen. Okay. And this hydrogen here is forming bond with this nitrogen, third nitrogen present. There is a hydrogen present here also. This hydrogen is nothing but the hydrogen of this nitrogen. This particular nitrogen is having the first nitrogen, and here there are two hydrogen again present, that is forming bond with this oxygen atom. This oxygen atom, here this bond is formed. Okay, so this is how the bond formation is happening. Okay, it may form, or it might form bond with this hydrogen also, this hydrogen, whatever it is, this NH2, one of the hydrogen of the NH2 molecule is sharing the bond with the oxygen atom. Okay. So this is how the three bonds are formed between the guanine and the cytosine bases. Guanine and the cytosine, the three hydrogen bonds are being formed, and in case of guanine cytosine, guanine is the purine base, cytosine is the pyrimidine base. And in case of the adenine and thymine, let me draw the thymine structure also. In case of thymine structure, there is present the thymine base, nitrogenous heterocyclic base. The thymine base is having the CH molecule again. The CH molecule is again having another CCH3, sorry, CCH3, and that one is again forming bond with the, having double bond, again forming bond with another carbon, and that carbon is forming bond with NH here, and that NH is having bond with C double bond O, which is again bonded with the nitrogen. This is what the thymine is. Okay, so the naming starts from here, the first nitrogen, second, third, fourth, fifth, and sixth. Here what happens, how the bond formation is happening in these cases. This nitrogen present here is having the two hydrogen, as I've mentioned, it is having two hydrogen, NH2. Now this NH2, one of the hydrogen will form bond with this oxygen, and this third nitrogen will form with the bond with the first nitrogen of the adenine, where there is hydrogen present. This hydrogen is nothing but the hydrogen of this nitrogen. Okay, so the first nitrogen present here for the purine base or adenine base is forming the hydrogen bond with the third nitrogen of the thymine base. Thymine is pyrimidine base, okay, thymine. Now this is how the bond formation is happening. So you can see there are 2 hydrogen bonds in case of adenine and thymine pairing, and there are 3 hydrogen bonds in case of guanine and cytosine pairing. Okay, this is what actually Watson-Crick's base pairing model mentions. Scientists Watson and Crick mentioned this base pairing model, that in case of guanine and cytosine there would be 3 hydrogen bonds, and in case of adenine and thymine there would be 2 hydrogen bonds. Okay, now this GC, the distance between these two strands of the DNA, that is the entire distance, this entire distance starting from this position to particularly this position, this entire distance is about 20 angstrom. This entire distance is about 20 angstrom in the the 20 angstrom in its in its 20 angstrom in its extended form or flat-like structure. Okay, in its planar structure, I must say. The distance between two strands is 20 angstrom. The optimum distance, and there is no less or more than this in case of this particular DNA. The type of DNA Watson-Crick first observed was known as the B DNA. Okay, this was actually the B DNA, because in case of B DNA, what happens is this strand which I have drawn in case of B DNA, there is 20 angstrom gap or thickness distance between the two strands. So 20 angstrom gap is there, and along with that, the distance between two nitrogenous bases, this particular distance is about 3.4 angstrom. Okay, 3.4 angstrom, the distance between two bases. And one complete turn of the DNA helix, is almost having 10 bases. So one complete turn is making up almost up to 34 angstrom. This is what Sir Watson and Crick has mentioned when they discovered the form of the DNA, and this DNA later on came to be known as the B DNA, because B DNA is having all these distances among their base pairs. Now there was another scientist known as Erwin Chargaff. Sir Erwin Chargaff, what he told is, this particular DNA molecule, a particular DNA molecule, in any kind of DNA molecule, generally whatever it is, whatever the species or organism it is, in that particular species, the number of adenine and adenine plus guanine bases that are present in one strand, one particular strand of the DNA, is equal to the number of thymine plus cytosine nitrogenous bases present in other strand of the DNA. Okay, this is the first strand and this is the second strand. Why? Because adenine is pairing with the thymine, right? Adenine is pairing with thymine and guanine is pairing with cytosine. So if the number of adenine is, suppose the number of adenine base pair in one strand is 10, then the adenine will pair with 10 base pairs, 10 thymines with present in another strand. So, and the guanine, number of guanine in one particular strand is suppose 5, then the guanine will pair with 5 cytosines present in another strand. So the total number of adenine and guanine, if we consider, then it would be, suppose if I write here, adenine is 10 and the guanine is 5. And so thymine will also be 10 and the guanine will also be 5. So 10 plus 5, 15. So adenine, guanine, total number of nucleotides, purine nucleotides is 15 in one strand, which is equal to the total number of pyrimidine nucleotides in another strand. So whatever, now it is not that one particular strand will only have the purine nucleotides or only have the pyrimidine nucleotides. One particular strand can have both the purine and the pyrimidine nucleotides also. But whatever the total number is in the polynucleotide strand, always the number of adenine and guanine, the purine nucleotides in one strand, is equal to the pyrimidine nucleotide is another strand. This is what Sir Erwin Shoggoff has told. He discovered. And another thing Sir Erwin Shoggoff has proposed, that is, in this particular, there is a particular value, okay, particular ratio of this base pairing. That ratio is known as adenine plus thymine divided by guanine plus cytosine. This is what the base pairing ratio is, that is the total number of purine and pyrimidine present in a particular DNA molecule divided by the total number of the guanine and the cytosine present in a particular in the same DNA molecule, because it is not always that if the total number of adenine, the purine bases are present 10, then the pyrimidine bases in that particular strand would also be 10. There might be more purine bases or pyrimidine bases, there might be less purine bases, or the reverse is also true, there might be less pyrimidine bases. So the number of adenine plus thymine, what the adenine is the purine base and the thymine is the pyrimidine base, okay, that are present in one particular strand divided by the number of the guanine and the cytosine present in that particular strand is having a definite ratio, okay, is always maintaining a definite type of ratio in case of all the species of organisms. And this was first mentioned or brought forward by scientist Erwin Shoggoff. Okay. So this may vary species to species. It may vary. The ratio of this adenine plus thymine divided by this guanine plus cytosine is almost 1.52 in case of the human beings. Okay. This ratio was found almost to be 1.52 in case of the human beings. Let me write it on the board. It is almost 1.52 in case of humans and 0.93 in case of Escherichia coli or E. coli. It is found that 1.40 is present in case of higher organisms or multicellular organisms and it is seen that it is about 0.60 present in lower organisms or lower group of organisms, okay, lower group of organisms. So basically what it is, this A plus T by G plus C, what is this thing basically? It is, as I've mentioned, that here it is like adenine and guanine, the total number of adenine and guanine, the purine base present in one strand should be equal to the total number of thymine and cytosine present in the opposite strand or the antiparallel strand, okay. This is antiparallel, see the 3' end is here, 5' end is here, and in this case 3' end is here, 5' end is here, okay, this is antiparallel. Now in this particular base pairing ratio, the number of adenine, it is why is this different? This is different because if a particular if the total number of adenine, okay, and the total number of guanine is being added and it is being divided by the total number of thymine and the total number of cytosine, okay, then the ratio might be 1 or might not be 1, because if we have the total number of adenine and guanine of a particular species and we divide it by the total number of guanine base pairs and the total number of cytosine base pairs, then that might not lead to 1 completely, because it would be not an ideal base pairing, because no species would have ideal base pair, that is the equal number of if I consider the entire DNA, not a particular strand, okay, if we consider the entire DNA, both the strands, if there are 10 number of adenine present, then 10 number of thymine present on the opposite strand. Okay, as per this formulation. But what are the number of thymine present in that same strand? We don't know. So in that case, if the total number of adenine and the guanine present on that same strand and is divided by the total number of thymine and the cytosine present in the same strand, then the ratio might not be 1, because it is not possible that the ratio is always 1 in that case. There might be variation. The purine bases might be more, the adenine might be more, the thymine might be more, but the guanine might be more, or the cytosine might be more, or they may be less. Either of them may be less. So it is very possible or obvious. So if we consider the total number of adenine plus thymine, that is adenine is pairing with the thymine, right? That's why it is adenine and thymine is being added, as per Sir Arvind Shargaf has mentioned. And now the total number of adenine plus thymine base pairs. Okay, suppose adenine is 10, then the thymine would be 10. Now, if we consider two strands, both the strands, the total number of adenine and the thymine base pair is forming the ratio with the guanine and cytosine. So, why the adenine and thymine is being added? Because adenine is pairing with thymine. So this is nothing but the base pair ratio, not the base ratio. That's why the pairing is being considered, and the guanine is pairing with cytosine. Now, in the same concept, if we proceed, that a particular DNA molecule is having certain number of adenine and thymine, okay, and that same certain molecule, certain species is having some more number of guanine and cytosine, or less number of guanine and cytosine. We are considering the base pair ratio including the entire DNA, the entire genome we are considering. Okay, so it is not that one DNA, one strand is having the equal 10 adenine, the other strand is having 10 thymine, and this particular strand here, the first strand, will not have thymine. It is not like that. This first strand might also have thymine and cytosine, the purine bases. Okay, so we are considering the entire thing. The adenine present in this strand and the thymine present in the second strand, the thymine present in the first strand and the adenine present in the second strand. Entirely, we are considering the either or case of both the strands and having this total adenine plus thymine base pairs. Similarly, the guanine and cytosine is also manifested or encompassed in this fashion. That is how the base pair ratio has been formulated by Sir Arvind Shargaf. And it is found such and such: 1.52 for humans, 0.93 for Escherichia coli, 1.40 for higher organisms, 0.60 for lower groups of organisms and all. Okay. So this is what Sir Arvind Shargaf told regarding the base pairing, and these three hydrogen bonds present over here, they require these three hydrogen bonds require more higher temperature in order to break the del G value of these three bonds. Okay, so this particular three hydrogen bond is known as the high melting area. High melting area, and these two hydrogen bonds require less heat for breaking, or less temperature for breaking the del G value. This is known as low melting area. Okay. Now, another fact is, what is this breaking of the bond? This breaking of this hydrogen bond is known as the denaturation, a denaturation of the DNA. So when these hydrogen bonds are being broken down, or the heat is given in order to break the pairing of the hydrogen bonds, that particular thing is known as denaturation. When the two strands of the DNA separate from each other, they start unwinding from each other, the helix breaks, the helix conformation totally disrupts. That is what denaturation is, and renaturation means the opposite. Renaturation means the formation of this hydrogen bond or realignment of the hydrogen bond again in order to form the helix combination or conformation. Okay. So this is what this is how the base pairing of the DNA structure is. The adenine, the purine structure, adenine and guanine structure, heterocyclic rings are a little bit complex and larger compared to the purine heterocyclic rings. This is the anti-parallel strand. 5 prime is here, 3 prime is there, and this is the parallel. This one is anti-parallel to that strand. This 5 prime is here and 3 prime is here. This particular strand is known as the leading strand. Okay. Leading strand. And this one is known as the lagging strand. And always remember, the base pairing is complementary. That is, this adenine is complementary with thymine only, and guanine is complementary with cytosine only. So the base pairing would obviously and definitely happen between two complementary bases. This is how the base pairing rule follows. This is what the base pairing rule follows. So, in order to maintain the rigidity or stability of the DNA structure, and this strand, this 3 prime, if we consider that one as the leading strand, then this would be the lagging strand. And sense strand means if we consider this as the sense strand, the 5 prime to 3 prime, okay, if we move in this direction and this is sense strand, then the complementary strand of that sense strand is anti-sense strand, that is 5 prime to 3 prime anti-parallel strand. So if we consider that one as the sense strand, 5 prime to 3 prime, if we move in upward direction, then this would be the anti-sense strand, 5 prime to 3 prime, if we move forward downward direction. Okay, this is what happens during the time of any kind of molecular marker technique. Okay, molecular marker technique in order to repair any kind of defect or abort any kind of process from the DNA transcription or translation, whatever it is. These certain tools and techniques are being adopted by the wet lab technology or molecular marker technique in order to produce an opposite complementary strand to suppress the function of the existing sense strand or the leading strand. And this is how the convention of the polymerization and the replication or the transcription of the DNA is happening. The central dogma is following. Okay. So this is what it is, that how the base pairing is happening between the two strands, and what are the formulations or theories that are being brought into the limelight by the scientists Watson and Crick and Sir Erwin Shoggoff for this particular purpose, in order to understand the polynucleotide chain in a very calculative and robust and radical sense, time effectively. Okay. So if you like this video, share the video as much as possible, subscribe my channel positively in order to get more such updates about this mind-boggling videos and all, and also comment down in the comment section below, hit the thumbs up button. Thank you.